I will be talking about some progress in the field of human brain imaging. Charting the functional regions in the human brain has been a long-standing aspiration in the field of neuroscience, marked by a rich and historical journey. For example, the exploration of language function commenced over 160 years ago with the seminal work of Paul Broca, who discovered that the left inferior frontal gyrus might be the speech production center of the brain. And in the 1960s, Roger Sperry's split brain experiments extended our understanding of language localization, underscoring the predominance of left hemisphere in the linguistic processing. From the 1950s through the 1980s, Penfield and Algerman used the cortical stimulation to further refine the mapping of language-related areas in the human brain with greater precision. In 1990s, the rise of non-invasive functional imaging technology, especially the functional MRI, has enabled a more accessible and more detailed mapping of the brain's functions, taking us a step closer to unlocking the mysteries of the mind. And beyond language mapping, functional neuroimaging has made significant contributions to neuroscience, leading to new discoveries such as the fusiform phase area, the default mode network, and more recently, the somato cognitive action network, and many more. While functional imaging has achieved great success in basic neuroscience, it's facing significant challenges before widespread clinical applications. Here I summarize several major challenges. First, there's a substantial individual differences in the organization of functional networks, which I will give you more details in a few minutes. Second, conventional functional MRI technology has very low reproducibility. It's only about 39%. Third, due to the correlational nature of functional imaging, most findings still lack causal validation. The challenge of individual differences in brain functional organization was recognized as far back as in the era of Algerman. Using invasive cortical stimulation, he found substantial variability in the locations of the language area among individuals. Our non-invasive functional MR results echo this significant variation. So in one experiment that I'm showing on the slide here, I had two students lie in the scanner, in an MR scanner, performing an hour-long language task. We can see that the student on the top, his language area was primarily in the left hemisphere, and the other student's language area is primarily in the right hemisphere. This variation could be even larger in the patient's brains. So to understand what regions are more variable across individuals than other regions, we use functional MRI to study individual differences of functional connectivity in a group of healthy individuals. We found that the primary cortices, which are responsible for the motor, sensory, visual functions, as shown. Oops. So as shown in the blue here, these primary cortices are demonstrating lower inter-subject variability. However, the primary, the frontal region, the parietal region, and the temporal cortex, which are responsible for the higher order cognitive functions, demonstrate substantial individual differences. And this pattern can be seen even in the newborn babies, suggesting that cognitive areas are already highly variable even at birth. And interestingly, we can see these similar patterns in the macaque brains. shown on the right here. So this result indicates that individual differences in the brain function may result from millions of years of human brain evolution. So in summary, individual variability is a fundamental property of the human brain, and neuroimaging technology should focus on revealing the the unique functional architecture of individual subjects before clinical application. To reliably map out the brain functions in the individual brain, we have developed a method known as personalized brain functional sectors mapping, or PBFS in short. This technique includes a robust and efficient imaging data processing pipeline and an iterative algorithm for identifying functional sectors in individual subjects. And using this approach, we can map out the unique functional network organization in individual brains with very high reproducibility. With the 30-minute MRI scan, while the subjects are simply resting in the scanner or even under anesthesia, we can map out 213 fine-grained functional sectors for each individual with the reproducibility up to 90%. While we are able to identify these 213 functional sectors, we do not fully understand the exact role of each sector. To chart the functional brain, we designed dozens of tasks and examined how these sectors participated in these different functional tasks. We then applied Bayesian algorithms to determine the exact functional role of each sector. So let's take the prefrontal cortex as an example. We found that the yellow sector played a predominant role in many cognitive control tasks. And the brown sector is a key player in working memory tasks. And the green sector is very important for attention, and the blue area is more for the declarative memory. So by labeling the exact function of these 213 sectors, we will be able to chart the function of the entire brain. We then carry out a series of studies to validate the accuracy of the PBFS maps. The most rigorous validation is to lesion the functional sector and test whether we observed expected functional impairments. So in one experiment, we recruited 42 epileptic patients who underwent neurosurgery. And before surgery, we map out the language network for every patient, and surgeons perform the resection blind to our mapping results. So for example, in this subject, the red area is the language network, and you can see that in the post-surgical MRI, this subject's language area was damaged. And among these 42 patients, we found that 20 patients exhibited post-surgical aphasia. In all of these subjects, the resection was too large and had lesioned the language area identified by PBFS mapping. And conversely, in the remaining 22 patients whose PBFS language network was not affected, none of them suffered from post-surgical aphasia. So these results support the accuracy of our language mapping. While our PBFS technology can assist pre-surgical mapping, its application may go far beyond neurosurgery. A recent progress is to guide brain stimulation using functional imaging. And brain stimulation has gained traction in the past 2 decades. For example, deep brain stimulation, or DBS, won Lasker Award in 2014. And transcranial magnetic stimulation, or TMS, is another promising technology which received FDA approval for treating depression and OCD. However, due to the difficulty of localizing accurate functional targets for stimulation, DBS is still limited to movement disorders today and cannot be used to treat other functional disorders such as depression or Alzheimer's. And for the same reason, TMS treatment is not personalized, but uses a fixed target for all patients, which has greatly limited its efficacy. So our previous research indicated that different neuromodulation methods, no matter is invasive or non-invasive, can be all effective if we hit the correct functional circuits. Therefore, if we can identify the functional circuits in the individual brain, then it's possible that, to design a personalized and non-invasive treatment strategy to achieve high efficacy. To test this hypothesis, we conducted a randomized clinical trial in patients with post-stroke aphasia. Using the PBFS, we localized language areas in each patient and identified a stimulation site within the language area, as indicated by the small dot here. And in this plot, each green dot represents the stimulation site of one patient. Noted that these stimulation targets can be several centimeters apart between different subjects due to the high individual variability. So after 3 weeks of non-invasive, personalized TMS treatment, we found that patients in the active treatment groups showed a response rate above 90%, whereas the control group, who only received the sham stimulation, showed a response rate of about 28%. And this plot on the left shows the improvement of language function in the 2 active treatment groups and the control group within 3 weeks. So I have shown you that our PBFS technology is able to delineate the landscape of functional networks. However, these functional MRI signals are not direct measures of the neural activity, but are very slow hemodynamic responses associated with neural activity. To date, we still lack an imaging technology that can capture the neural activity in the living human brain with both high spatial resolution and high temporal resolution. As shown in this figure, existing tools all confront a trade-off between spatial resolution and temporal resolution. And the ideal imaging technology should sit at this corner with high spatial and temporal resolution at the same time. And last year, a Korean group published an astonishing paper in Science in which they introduced a method to directly measure neural activity in a rat brain using MRI. The method they called Diana seemed to overcome the trade-off and achieved high spatial resolution and temporal resolution at the same time. However, many groups attempted to replicate this study and failed. The primary challenge lies in the fact that MRI signal changes associated with neural activity are extremely subtle. To address this challenge, we developed a new MRI sequence, which we call Trigger. Compared to Diana, our sequence features a larger flip angle and a relatively longer repetition time, resulting in an improved signal-to-noise ratio. As shown in the middle figure, the Trigger sequence demonstrates much higher SNR, which is shown in red, compared to the Diana, which is in blue. And most importantly, by shortening the interval between 2 consecutive echoes, we can achieve a high temporal resolution of 1.4 milliseconds. So we then test this trigger technique with a passive visual task in awake human subjects. So the subject is only viewing a visual stimuli in the scanner. As shown in the left panel, significant trigger responses were observed in the early visual cortex, approximately 50 milliseconds after the stimulus onset. The time course of the visual cortex is shown on the right. To test whether the trigger response capture the neural activity at the millisecond scale. We delayed the visual stimuli onset by 10 milliseconds and found that the triggered responses were also delayed by 10 milliseconds. We then further delayed the stimuli onset, and the trigger signal also shifted further in time. So this suggests that our signal is really captured something with a very high temporal resolution. So we replicate this visual study in 3 independent groups of human subjects. Each group has 10 subjects. We can see that the trigger response can robustly track the delay of stimulus onset in each group, suggesting that it has very high spatial and temporal resolution. To further test whether the trigger signal reflects electrical activity, we performed the classic frog leg experiment in MRI. We know that electrical stimulation on one side of the spinal cord will cause the contralateral thigh leg to contract. Because this experiment does not involve changes in blood oxygen, noted that the heart has already been removed in the frogs, so we can rule out the potential influence of hemodynamic responses on our trigger signal. So as shown in the right panel, we use subthreshold electrical stimulation to the spinal cord, which induces significantly stronger trigger signal in the leg, as shown in the red, compared to the non-stimulation condition, which is in blue. So this result is from 10 frogs. So these results together indicated that our new trigger technique holds the potential to directly record neural activity with the millisecond resolution. So to quickly summarize, I have shown you today that our PBFS technology can identify functional fingerprints of individual subjects with a relatively easy procedure. The subjects only needs to stay in the scanner for 20 to 30 minutes at rest. And we can use these PBFS maps to guide personalized neuromodulation. We have been conducting a series of clinical trials in depression patients, in autism patients, and so on. And we have been pushing our imaging technology forward to directly record neural activity in the living human brain. So these technical advances will provide us useful tools to study the neural underpinnings of cognitive processes and may lead to personalized treatment strategies for a variety of brain disorders. And lastly, I want to thank the members of my team. I feel very lucky to work with a group of very talented young scientists. And I also want to thank the support of Champion Laboratory. Thank you very much.
All right. Thank you, Hesheng.